Hello. Today we will see a video lecture on sequence data formats. I'm Dr. Anand. Let's get into the topic. First, let's see introduction to biological data formats. Biological data formats define how biological sequence structure and annotations are stored and processed. The importance of data formats include they enable data sharing and interoperability, supports bioinformatics tools and pipelines, ensures efficient storage and retrieval. Biological data formats are essential for computational biology, allowing research to analyze genomics, proteomics, and structural information effectively. There are various biological formats one has to understand. Let's see what are the key biological formats that we will be covering up today. So now, having seen some key sequence formats, let's move on to a very important structural data format called as PDB. PDB stands for Protein Data Bank format. It's a text-based format for storing 3D information data of biological molecules. The usual PDB structure involves a header, title, compound information, series informations, atom information, heteroatom information, connections, that is the bonding information, and end of the file. So here is an example of the PDB where you see the header information, the title information, the atom information involves the XYZ coordinate system with the atom information. Then the heteroatom information here, what heteroatom is present among this, and then the end of file. So the key features involve stores atomic level structural data, essential for molecular modeling and docking, supports protein ligand interactions. Applications of PDB include drug discovery, protein engineering, structural biology, and you can access PDB information from rscb.org. Now let's go to next format called as GenBank format. So GenBank format is a very commonly used format by NCBI for storing information. So it's a rich annotation sequence format used by NCBI. The basic information structure involves locus, features, and sequence, and used for genome storage and retrieval. So here is the locus information, definition, accession, keywords, references, metadata associated like source, gene, and CDS, and then the sequence information itself. Now let's see some feature information formats like GFF and GTF. So GTF and GFF are similar as a fixed structure mainly used for RNA sequence analysis. So the usual structure involves chromosome, source, feature, start position, end position, scores, strand, frame, and attributes. So the key difference between GFF and GTF are GTF uses quotes, whereas GTF uses gene ID. Okay, and GTF explicitly defines gene ID and transcript ID. GFS is more flexible, while GTF is having a stricter data format. A very good annotation format for RNA studies. Now let's see another important genomic format called as VCF format, because this VCF format speaks about variants, that is SNP information. So variant calling format is usually used for storing genetic variations such as SNP, SNP stands for single nucleotide polymorphism, involving insertions, deletions, and structural variations. It is commonly used in genomic studies and NGS analysis. Usual VCF structure involves three main sections, that is the metadata, header, and header information, and data section. So under header information you have all these chromosome position, ID, ref, alt, qualifiers, everything, and under the data section you have each row representing a variant, like say chromosome 1, 2, 3, 4, 5, RSID is given, then what position like A, G has been translated, and then the other metadata associated with the variant. Now that we have seen all the sequence format, let's see another important sequence format that is used for phylogenetic studies called as NIVIC format. NIVIC format represents tree structure using nested parentheses. It is a widely used phylogenetic analysis. It is widely used phylogenetic format used in tools like Mega, Phylab, Phylum, and Beast. So basically, the A, B, C, D in this format represents species or taxa, and then 0.1, 0.2, 0.3, 0.5, etc. represents the branch length, okay, of the or the evolutionary distance. So this is separated by parentheses indicating the groupings or the clades. So the colon dot colon marks the end of the colon marks the end of the tree. So this format will usually generate a tree that is represented here at the bottom. So this comes to end of key biological data formats. Thank you for your patient listening. Thank you.